If you cut them in half, the bottom end grows a tail, the top end grows a head. But these cells that you just separated were next door neighbors before you cut them. How come they have completely different anatomical fates? That question cannot be answered locally. You can't tell from where you are what you should build. You have to communicate with the rest of the body and figure out what's missing. So this is non-local decision making. So we asked this question: when you amputate this planarian, how do you know how many heads you're supposed to have? And it turns out that there's an electrical gradient that we characterize that tells you how many heads you're supposed to have. And if you and what you can do is you cut off the head and the tail, and this middle fragment will regrow a nice perfect worm 100% of the time. But because we understood this gradient, we were actually able to make these two-headed worms by taking this electrical. So here it says one head. But now we can say, no, actually you should have two heads. And sure enough, this isn't Photoshop. These are real animals. They make nice two-headed forms. Now, here's the really important part. This bioelectric map is not a map of this two-headed creature. This bioelectric map is a map of this one-headed creature. So what we did was we rewrote the the stable bioelectrical pattern memory in this one-headed animal, and it stays one-headed with a normal gene expression. So here, anterior genes only in the head, not in the tail. And it sits there perfectly happy like this until it's injured. So a single planarian body is able to store at least two different representations, two different ideas of what a correct planarian looks like, and sit there, and it's a latent memory. It doesn't do anything until you injure the animal. The same body, the same one-headed body can store two different representations. So at the very beginning, I told you that this collective intelligence works towards, it's a homeostatic loop that works towards a specific set point. And the question was, how do you store a set point for anatomy? This is it. You are literally looking at an example of how of how pattern memories are stored. They're stored as a stable bioelectrical state in tissue. And it's this in particular is kind of cool. It's a counterfactual memory because this is not a representation of what's happening now. It's a representation of what you're going to do in the future if you get injured. This is memory. It's long-term stable. It's rewritable. It has conditional recall, as I just showed you, or latency, and it has discrete behaviors. So what we're doing is we're reading and writing patterns into the collective intelligence of these cells that must navigate anatomical space. And so now what that means is that we can really do the kind of computational work of fleshing out the connections between the electrical state space of the circuit that we understand with all kinds of ideas from connectionist computational neuroscience to understand how electrical networks can store patterns, do pattern completion, and navigate spaces intelligently.